Okay, so this is the first set of checkpoint questions. And let's just discuss these here. Okay, so what are the male gonads called? This was the testes. What are the female gonads called? This was the ovaries, ovary or ovaries, testis or testes. The second half of the question, besides gametogenesis, what is the gonad's other primary function? So the gonads had two functions. One was gametogenesis, the growth and development of the sex cells, the gametes, which are the ovum and the sperm or spermatocyte. And then the second was hormone production. So the production of hormones, estrogen, progesterone, and androgen, such as testosterone, that would help to sustain those gametes, okay? The second question, what is the distinction between sex determination and sex differentiation? So sex determination is essentially genetically determining the sex of the embryo. This is based upon whether or not there's the Y chromosome present and whether or not that accurately presents the SRY genes, which are the sex determining region of the Y chromosome, and then whether or not the genes present coded for the testis determining factor and began to produce testes. So that's kind of the first half of the equation that takes place within the first few days of implantation and embryonic development. Then there was sex differentiation. So sex differentiation was completed by the second week. This is where we determine the sex of the embryo phenotypically. So whether or not we have development of the male sex organs, such as the testes, and then the other accessory organs, the prostate gland, the penis, etc., the vas deferens, or we have the development of the Müllerian structures. And the Müllerian structures we said were the uterus, the vagina, the fallopian tubes, etc., etc. So that was determined by whether or not the testes were there primarily. And if the testes were there, they would secrete testosterone and Müllerian inhibiting hormone, MIH. And if those two hormones were being secreted, then we would have the progression of the Wolffian duct structures and the regression of the Müllerian structures. And if those two hormones weren't there, then we would have the default option, which is the Müllerian structures developing into the female physiology, the female anatomy rather. Okay, so that pretty much answered question 2 and 3, which is what were the deciding factors. Alrighty. Okay, let's look at the other set. I think we had some more at the end of this packet. Okay, here we go. Okay, so question 1. For each of the following descriptions, indicate whether it applies to Sertoli cells or to Leydig cells, which are the two main types of cells in the testes. So are present in seminiferous tubules. Those are the Sertoli cells, the cells that actually line the tubule that we call the seminiferous tubules. Are present outside seminiferous tubules. Those are the Leydig cells. So these are the cells found out in the periphery of these tubules, out in the interstitium, and hence we sometimes call them interstitial cells. Secrete testosterone. Those are the Leydig cells, right? Remember, the interstitial cells are the ones that secrete testosterone. And then are the target cells for FSH. Those are the Sertoli cells. So the actual cells that are helping with spermatocyte development, that are lining the tubules, that are creating that basal surface and that laminal surface where the actual spermatogenesis is taking place. Those are the cells that are intimately involved in that. The next feature here are target cells of LH. So those are going to be the Leydig cells. Luteinizing hormone stimulates Leydig cells to secrete testosterone. And then finally, secrete inhibin. Those are the Sertoli cells. Okay. So the Sertoli cells had two functions, spermatogenesis and sperm development, as well as secreting inhibin, which is a negative feedback onto the pituitary, the anterior pituitary. And then the Leydig cells had one main function, which is secreting testosterone, but testosterone had three functions. Negative feedback onto the hypothalamus, negative feedback onto the anterior pituitary, specifically LH secretion, and then supporting the Sertoli cells in that sperm development, right? So testosterone acted on Sertoli cells as well. Okay, I hope that was clear, not too much of a confusion. All right, question 2. Arrange the following structures so that they correctly describe the path of sperm transport from the testes to the penis. And then these are the structures that are encountered. So the correct route is going to be from the seminiferous tubule where they're created, alongside the tight junctions of those Sertoli cells, then to the rete testes, which is kind of the culmination of the seminiferous tubules, then into the efferent ductule, which is heading towards the epididymis being that prominent region on the side of the testes. From the epididymis, they're stored in the vas deferens. Remember, they're kind of sitting there until the sexual response is elicited. And then when the sexual response is elicited, they travel to the ejaculatory duct and make their way into the urethra. Okay, at this time, they're going to be joined with the other fluids, and this is where the secretion is called semen. Alrighty, question 3, describe the role of the autonomic nervous system in the male sexual response. So here we said the first phase, which is erection, controlled by the parasympathetic system. The second phase, emission, controlled by the sympathetic nervous system. And then the third phase, ejaculation, was also the sympathetic nervous system. Okay, and erection, emission, ejaculation in that order. Erection was the stimulation, so depending on the mechanoreceptors in certain erogenous zones. The emission was the traveling of the sperm from the vas deferens, where they're kind of waiting, into the urethra, and then ejaculation was deposition of the semen at this point into the female reproductive tract. Okay. All right, so I feel like those checkpoint questions really kind of summarize the important points that we